All right, class. So now what we will do is we will zoom into this part. Okay, we will zoom in, blow it up, and we will see how certain molecules are transported across the simple cuboidal cell of the proximal convoluted tubule into the interstitial space and then ultimately into the capillary. So we will look at this route in details. Okay, so here is my representation. What I have drawn is a simple cuboidal cell of the proximal convoluted tubule in black. And this cell, this is the apical surface of the cell. That's the apical surface. And this is the basal surface. Okay. And the apical surface has microvilli. Why do you think we see microvilli on these cells? Because these cells will absorb things, right, from the lumen into the interstitial space, and therefore having microvilli will increase the surface area for absorption. In this simple cuboidal cell of the PCT, we also see a black colored nucleus. Then at the bottom, we do see an endothelial cell that lines the capillary. And in between the simple cuboidal cell and the endothelial cell, we have a space called interstitial space. So let's play. Let me see if I can change color. Okay. So here we have this transporter, and this is called SGLT transporter. This transporter transports sodium from the lumen. This is the lumen, so there were a lot of sodium floating around. So sodium is transported across the membrane via the SGLT transporter. So now we have sodium inside, right? Now sodium cannot stay in the cell forever. At the basal surface right here, there is a sodium-potassium pump. This pump will get rid of 3 sodium and will take in 2 potassium. The potassium that comes in will eventually leak out into the interstitial space by a potassium channel. So this one is a open potassium channel, also called a leak channel. All right, very good. What else comes in along with sodium through the SGLT transporter? Well, we have glucose coming in. So we have a lot of glucose in the lumen, right? The glucose comes in through the same transporter. Now, obviously, we cannot keep too much glucose in the cell. The glucose needs to be transported to blood so that it can go to other cells of the body and be utilized to generate energy. So here we have these special transporters called GLUT. Do you remember GLUT, the GLUT transporters? So we have these GLUT transporters that will carry the glucose to the interstitial space. Okay. What else? So we are actually picking up both sodium and glucose from the lumen into the simple cuboidal epithelial cell, and we also are transferring the sodium and glucose into the interstitial space. What else? Well, as sodium and glucose comes in, have you forgotten water? Osmosis, water will follow. So we will have water coming in, and then this water will move out by diffusion, by osmosis, sorry. All right. So now we have sodium, we have glucose, and we have water in the interstitial space. These things will move into the endothelial cell of the capillary by simple diffusion. So glucose will enter the cell by diffusion. So will potassium. So potassium and glucose enters the cell by diffusion, and then water enters by osmosis. So water enters by osmosis. All right. So this is how things get reabsorbed at the proximal convoluted tubule from the lumen into the capillary endothelial cells. So let's take a look. This is something that I have previously drawn out. Let's go over this one more time. We see the lumen where we have a lot. We will go from top to bottom, okay? So on the top, we see lumen, and we do see a lot of sodium and glucose in the lumen. We see the simple cuboidal cell with microvilli drawn in black. This cell also has a nucleus. And then at the bottom in red, we see capillary endothelial cell. So sodium will be transported into the cell from the lumen by SGLT transporter. The sodium will then leave the cell by sodium-potassium pump. The sodium-potassium pump will allow two potassium ions to enter the cell, and these potassium will then leak back into the interstitial space via the open potassium channels. Along with sodium, glucose gets co-transported into the cell, and this glucose then leaves the cell via specialized GLUT transporter, which are located at the basal surface of the cuboidal cell. As sodium and glucose are coming into the cell, water follows. So water comes in by osmosis, and water also leaves the cell by osmosis. Now we have a lot of sodium, potassium, glucose, and water in the interstitial space. Of course, they cannot hang out in the interstitial space forever. So potassium and glucose will enter the endothelial cell by simple diffusion, and water will enter the endothelial cell by osmosis. And here is a picture. So here I have written down everything. So in this picture, let's focus on the picture. Right here, you see the co-transporter. This is the sodium glucose co-transporter. We can see both sodium, shown by the purple balls, and glucose, shown by the blue balls, entering the cell. Sodium then exits the cell by this sodium-potassium ATPase. That's the sodium-potassium pump. And as sodium leaves, potassium enters the cell. We see this. This potassium will then eventually leave the cell by a leak channel, not shown here. The glucose that enters the cell, as shown in blue ball, will then get out of the cuboidal cell by facilitated diffusion across the GLUT transporter. Eventually, we will have potassium and eventually we will have glucose entering the capillary by simple diffusion. Same will happen for potassium and also sodium. So everything moves in and out of the capillary by diffusion. All right. Okay. So moving back to our second lecture, we did talk about a concept called transport maximum. Do you remember that? So what is that? So remember that when we are looking at facilitated diffusion, we are always looking at these proteins which can hold hands of molecules to help it get transported across a membrane, right? So let's say we have a membrane here, and then there are these transporters. They will transport molecules from this side. They'll bind to these molecules and transport them to this side, right? However, can we overwhelm these transporters? Yes. If we have too many molecules that are on one side and all of them need to come to the other side, of course that will overwhelm the number of transporters. That brings us to the concept of transport maximum. So the concept is that transport rates increase with the increased molecule concentration until saturation is met. This is transport maximum, where all carriers are in use. So we have talked about this before. So let's look at the graph. In this graph, on the x-axis, we have concentration of X, and on the y-axis, we look at the rate of transport of X. So as the concentration increases, the rate of transport increases. That's okay. However, at this point, there is a problem. All the transporters are saturated. Therefore, no matter how much the concentration increases, so even with increase in concentration, there cannot be any further transport. So the transport kind of plateaus here. Because the transporters are saturated at that point. And this point where the rate of transport cannot be further increased by increasing the concentration of the substance, that is called transport maximum. We will apply the same concept and see whether concentration of glucose has an effect on a different transportation, whether glucose concentration affects its transportation across the membrane. So we are going to look at the relationship between the plasma glucose concentration, which is plotted on the x-axis, and we will look at how the plasma glucose concentration affects the filtration rate, the reabsorption rate, and the excretion rate of glucose. Okay, so on the y-axis, I have plotted the filtration rate of glucose in yellow, the reabsorption rate of glucose in purple, and the excretion rate of glucose in blue. Now, I want to talk about something first. In which part of the nephron will we have filtration of glucose? At the glomerulus, right? Glomerulus is a site for filtration. And remember, glomerulus, things get filtered out non-specifically. Whatever fits through the pores, whatever fits through the fenestrated capillaries, they can come out. So the point is, if there is a lot of glucose in the blood, in the plasma, a lot of glucose will be filtered out into the Bowman's capsule across the fenestrated capillaries of the glomerulus. So we see an exponential growth, or a curve that is growing exponentially. Increase in plasma glucose concentration will lead to more filtration rate of glucose at the glomerulus. Now we will compare the reabsorption rate of glucose. Now, where in the nephron does reabsorption of glucose take place? Yes, if you say proximal convoluted tubule, you are right. At the proximal convoluted tubule, we will have glucose reabsorption, and we just saw that, right? We just saw how these transporters are carrying glucose back into the blood capillary. All right, so the point is, think about the transporters. The point is, if you increase plasma glucose concentration, of course, there will be more glucose filtered into the filtrate, and then there will be more glucose reabsorption into the through the proximal convoluted tubule, but that will continue up to a certain point. At that point, after a certain time, if you have even more glucose floating in the filtrate, the transporters are busy working. So the transporters cannot help these glucose molecule to be transported back into the system. Therefore, you will end up reaching a state called transport maximum. So this is transport maximum for glucose at this point. And eventually, this will lead to excretion of glucose via urine. So let's look at the blue line now. So as the plasma glucose concentration